give rise to other types of brain cells. Not blood cells, not liver cells, but brain cells. And they have only a limited ability to generate more of themselves. It's not indefinite. And so in some cases, those stem cells will expire early in development of the fetus. In some cases, they'll last a lifetime. It's just tissue specific. I'd like to then talk about embryonic stem cells. And these are the ones that are the most controversial and that we've spent a lot of time talking about leading up to Proposal 2. Embryonic stem cells are derived from 4 to 5 day old, what we call blastocysts. And this is an example of one shown here. This is magnified many hundreds of times. But in reality, if you would look at this, to give you a concept of how big it is, it would fit inside the period at the end of a sentence. And it's this 4 to 5 day old blastocyst from which we derive these cells inside here. And then after undergoing some expansion in the laboratory setting and quality control checks, ultimately become a line of embryonic stem cells that we then use to make different cell types. And as Ed so nicely mentioned, that I will only just briefly review, is that these embryos, these blastocysts, come from those that are in excess of those or unsuitable for transplantation, only from IVF clinics. No money has changed hands. The couple has voluntarily consented, written informed consent to donate these without the exchange of money. The unique thing about embryonic stem cells and where they differ compared to adult stem cells is that they have a nearly unlimited ability to regenerate themselves or undergo self-renewal. In a laboratory setting, once we establish the line, they'll go on and on and on. It's a lot of work to keep them unspecialized, but they can do it for a long time. Additionally, these embryonic stem cells will give rise to the 200 or so different cell types in the body. Of course, they have to be exposed to specific chemicals to induce them into a specialized cell type, but each of these cells has the potential to become any of those 200 cell types. They're limitless in that capacity. What we have now, though, are another type of stem cell, similar to embryonic stem cells in some ways, and they're called induced pluripotent, or iPS cells. And these cells are actually derived from non-stem cells. So follow me here for a second. Literally, they can take a sample of skin from a patient, a donor, take that to a laboratory, expand those cells, and using molecular techniques that we have in the lab, which is the exposure to anywhere from 1 to 4 genes, when those genes are put into that skin cell, in a rare number of those cells, those genes can cause them to become embryonic stem cell-like, or iPS cells, such that 0.1 to 1% of the cells that acquired that iPS identity now have a nearly unlimited ability to regenerate or self-renewal and to give rise to 200 or so cell types in the body. There are some caveats to that, and I'll get into that in a little bit, but this is certainly a new technology that's interesting for a lot of reasons, but does have a few drawbacks. And so, to give you a little more depth into the uses that of stem cells that I talked about when I started my talk. Stem cells can be used to understand early development and differentiation, such as creating disease-specific stem cell lines. iPS cells are very useful for this. For example, say we have someone who suffers from Huntington's disease. We could take a sample of that patient's skin, take it to a laboratory, put several genes into that cell that then allow it to become an iPS cell. Now we have a model, a potentially useful laboratory model of early development of Huntington's disease with a specific mutation found in that patient's DNA. We can use stem cells to treat human disease or injury, and that's through the transplantation of cells that we engineer in the laboratory from the stem cells. And we can do that to treat cancers of the blood system, which is already ongoing, and I'll elaborate a little bit more on that as we go along. Parkinson's disease and diabetes are very kind of exciting targets for stem cell therapy, because those two diseases in particular are due to the loss in general of a specific cell type in a specific location. So Parkinson's, there's a specific area of brain cells that die off in a very specific location. So in a laboratory, we could take the unspecialized stem cells, push them to become the cell type lost in Parkinson's, transplant them back into the exact region, have them secrete the right chemicals, form the right connections, and improve or maybe cure the disease. And in animal models, there's proof of principle for that very concept to happen. They've demonstrated it numerous times in numerous ways. And the third aspect of how stem cells may one day change medicine is using them to screen drugs, to assay for efficacy or toxicity, whether they're wild-type cells, normal cells, or unhealthy cells. And so to give you some specific examples of how this happens, I want to talk about ways that each type of stem cell is used or can be used to benefit mankind. The first are adult stem cells. So we've known about we being scientists and physicians, have known and studied about adult stem cells for the last half century. And over the course of that half century, we've developed treatments for several diseases, most notably blood cancers like leukemia and lymphoma, through the process of bone marrow transplantation. We know that inside your bone, in the marrow cavity, the hollow space, exist blood stem cells, or those cells, stem cells that give rise to the blood system and immune system. And in some patients who are suffering from leukemia and lymphoma, we can take bone marrow stem cells from either themselves or a genetically related patient, transplant them back in, and treat and or cure that disease. In fact, that was a technology developed back in 1969 after about 14 years of trials, unsuccessful trials in patients. And the treatment is still far from perfect. We are modifying that as we go, but it's one aspect in which adult stem cells have borne fruit in terms of treating human disease. This one I just added yesterday is the neural stem cell transplantation for amyotrophic lateral sclerosis, or Lou Gehrig's disease as it's more commonly known. A group of clinicians and scientists, which will be led by Dr. Eva Feldman, is now undergoing the first trials to treat ALS using stem cell therapy, such that they can put these cells back into the patient and have them either reduce or prevent the progression of the disease. We'll see how it goes. At this point, the first trials are only safety. Is the treatment safe? From there, they go on to see whether or not it's effective. But nonetheless, another potentially promising treatment derived from stem cells, in this case, adult stem cells. Embryonic stem cells, we've only had the technology to really study them for the last decade. In 1998, a laboratory in Wisconsin developed the technology to culture human embryonic stem cells. And just this year, I think January of 2009, the FDA, the Food and Drug Administration, which needs to give the green light for for drug trials to begin, gave permission to a company called Geron to use human embryonic stem cell derived cells to treat spinal cord injury. So in the case of Geron, the name of the company that's starting this trial, they've taken embryonic stem cells and specialized them into one of the supporting cell types that insulates the nerves that go up and down the body. And what they want to do is see whether or not these supporting cells will help either lessen the symptoms or restore some function to people who have undergone spinal cord injury in 7 to 14 days before treatment. Currently, though, this clinical trial is on hold while they review some more information. But it's an exciting landmark application in the sense that it's the first clinical treatment, or clinical trial, I should say, of a cell derived from a human embryonic